All right, welcome everybody. It is Friday morning, and usually we would be having a lecture right now, but I am not on campus. I'm actually here at a Top Hat conference, and I thought I would want to take some time to draw a couple of figures to help you for your exam coming up on Monday. So, welcome to a special travel edition of Figure It Out. So I'm actually taking a break before I actually get some breakfast at this Top Hat conference, but I almost didn't even make it here. If you know, on Thursday morning it iced really badly. A lot of flights were canceled out of Philly to get here, but I ended up sitting on the tarmac getting the plane de-iced for about 30 minutes, and then everything was fine. So we made it. We're here at the Top Hat conference. I've gone to a couple talks, talked with a couple of people to try to improve the experience that you're all going to have in class. But before I continue on with day 2 of this conference, I did want to take some time to draw a couple of figures out for you. We're actually going to start off with mitochondria, then we're actually going to look at how we do the electron transport chain, what happens with oxygen, what happens without oxygen. So let's turn it around, put it on a tripod, we'll talk about it. Okay, so what I really want to focus on in this drawing is looking at what happens with the electron transport chain and the ATP synthase. Right? How do these things kind of work together? And to give us a reference of what we're looking at, remember we're looking at some of the machinery that you find inside of each and every mitochondria. So if a mitochondria is going to look like a smooth outer layer, we're going to have folded inner layers everywhere. And it's all along this inner layer we're going to see a bunch of electron transport chain and a lot of ATP synthase. Yeah? Okay, so we're going to focus in on just one region of this folded inner mitochondrial membrane to actually see how these proteins are going to work together. First step, let's draw this membrane. It's going to look like a cell plasma membrane, but remember it's not. It is this inner membrane that's right here. And I'm usually just going to draw this fairly quickly to represent a phospholipid bilayer that we're all kind of familiar with. So we're going to have something here called the electron transport chain. And the easiest way for me to kind of show you what this looks like is maybe to draw it like that. Here's our ETC, right? Embedded sort of right next to it. We're going to have our ATP synthase, which I'm just going to draw like that. And so how do these two things work together? So we have to understand what happens in the electron transport chain. It's actually made up of 4 to 5 different proteins. And what ends up happening is we take things like NADH, right? These reduced coenzymes. We have to break that bond, release that electron, and that electron then gets put onto this protein in the electron transport chain. What's its job? It finds an H plus proton, we call this the same protons, it pumps it out into this side. So if we look at our mitochondria, we're pumping them out to this side. Okay, why do we do that? Why do we want a whole bunch of H pluses? Well, we kind of already talked about the fact that we want H pluses here to act like a power source. That power source then being a flow of these protons back to the inside, right? Back into the matrix of the mitochondria. And when that happens, we can actually do things like make ATP. So here is a DP, right? Adenosine diphosphate. We really want to make it into ATP, adenosine triphosphate. So all we really have to do is we have to find a phosphate. We have to add it on, and then we get ATP. Now this is going to be an active process. It actually needs some energy, and again, this is going to be our source of energy. Okay, so this is where we left off in class. What we really have to discuss before your exam is what happens when you have oxygen. Right? Oxygen actually allows this to continue to go. Why? Because as the electrons are basically passed along the electron transport chain, all of its energy is used up. And at the end, you basically have a dead electron. You have an electron that is 0 charge, that has no energy. So what are you supposed to do with that electron? Well, you have to take it away from the electron transport chain. And the way that you do that is with oxygen. Oxygen grabs that last dead electron and it actually makes water as a byproduct, right? So that's really interesting. This is what happens when you actually do have that oxygen present. But what happens when you don't have that oxygen present? This usually happens when you are doing something that's really metabolically active and you just use up the majority of oxygen you have in your bloodstream. So what do you do at that point? Let's get a new piece of paper and draw this one out. Let's briefly talk about what happens when you don't have enough oxygen, right? What happens without oxygen? And to really understand this, we have to zoom out a little bit, and like to really understand cell respiration, we have three parts, right? We have glycolysis, right? Glycolysis. We're going to have the citric acid cycle / the Krebs cycle, and then we're also going to have the electron transport chain that we just covered, plus the ATP synthase, right? All of that working together. So the thing is, because these are in the mitochondria, if you don't have oxygen, that's going to shut down the electron transport chain. It's basically going to shut down all of the functions of the mitochondria, which means even though we want to use this to get a bunch of ATP, without oxygen, we can't. No problem. We can still make ATP somehow, right? Because glycolysis happens outside of the mitochondria. So even if this shuts down, we still have this method of breaking down glucose into pyruvic acids. We don't really care about the pyruvic acids. What we really care about is in this process, we can make ATP. Okay, so in this case, glycolysis becomes really important without oxygen because it becomes the only way you can make ATP. With oxygen, kind of the most important part are the pyruvic acids so that we can transfer them into the mitochondria. But if we shut this down, this becomes the sole way that we can make ATP. Here's the problem. What do we have to do the ATP, right? We not only have to break down the glucose, we have to put in energy to get some energy out, sure. But one of the most important things we see is having these NAD pluses, right? These oxidized coenzymes actually have to come in and get reduced, right? This is an essential step of glycolysis where we strip one of the electrons from the glucose, and we do this because we can actually bring this into the mitochondria, right? But if this shuts down, we can't do this, right? If this shuts down, we actually make a whole bunch of NADH that essentially is useless, right? These are just like full Amazon trucks that have nowhere to go. And when this happens, we run out of NAD+, right? Here's the problem. If you run out of NAD+, you can no longer break down glucose. If you can no longer break down glucose, you can't make ATP. So what is the solution that we use, right? You might have heard of like lactic acid. What we actually make is something called lactate. And this is a very temporary molecule. What it is it's a modified pyruvic acid. So we actually change this a little bit. But in this change, we turn the NADHs back into NAD pluses, right? So again, the reason why this is important is because at this point, without oxygen, all of this stuff is useless. All of this is useless. And we run out of NAD plus to continue to do glycolysis. So we convert this so that we can get NAD plus back and continue to do glycolysis and continue to make ATP. Now, eventually you're gonna recover. Once you have enough oxygen in your system, the lactate will basically re-enter your system and be able to be transformed back into pyruvic acids that brought into the mitochondria where you can continue to do this. But this is really what happens when you don't have enough oxygen. All right, hopefully that was helpful. I know that I just kind of drew stuff out on pen and paper, a little bit old school, but I think visually seeing this really helps. You know, not just looking at an image from a textbook or looking at an image that you can get from Google. I really recommend that you try drawing this out, or at least try drawing it out as I draw it out, because I think that's an easy way to get all that information. All right, I'm gonna go try to get some breakfast before this conference picks back up again. If you have any questions, please put them into our D2L question form and I will be happy to answer or add on to this video. But best of luck, have a great weekend. I will see you all on Wednesday for the exam.